in this video I will show you how can you annotate your variant calling file after sequencing. So this is a useful user-friendly tool that can actually tell you which mutation is a driver mutation and which mutation is a passenger mutation, which is very important to know in cancer studies. So I have a VCF file that actually looks like this. If I open the file, so basically VCF file has two sections. The first section will tell you how the sequencing carried out filtering of different variants, and then the final variants, which are your mutations, are actually a good quality mutations that you can trust. For example, if you see here in the filter ID, some variants didn't pass the filters because the reason could be indicate the variant has amplicon bias, and some of these variants has low depth and the DP was very low. That's why the variant didn't pass the filters. Low mapping quality. So all these filters have been mentioned why your variant has not passed the filters because of all these filters. And if you go down, you can actually see in this section that which tool they have been used to generate a VCF file. For example, this getk command line they have used, and also they have used we can check here they have used filter mutex calls as well to generate the final VCF files. And if you see here, um, now in the info section they will tell you what each abbreviation means in this file. For example, the allele frequency, and if you go down in this file, all the information which is present in this file, and this file will give you all the details, all the abbreviations. And here when you come down here, they will tell you which reference genome has been used to align all the reads. So basically we used HG19 reference genome for our mutations and for our tumor samples, and here they will tell you the name of the tumor sample that you have run. And also in this section, this is the mutation section. So basically this section will tell you on which chromosome your mutation is present, and also the exact position on the chromosome, and the reference base and the altered base in the mutation, also the quality of your mutation and which filter section. You will see different descriptions whether your mutation has been passed the filter or not. So in my case, all my mutation has been passed all the sequencing filters. If my mutation wasn't past the filters, something like this would be written in front of it, like a reason why you should not trust this mutation. For example, the low map quality, so you have to filter out only the pass variants. So in my file I already generated a final file for myself where I only have the pass mutations in my file. So now I can use this VCF file to basically annotate my variants. So I will close this VCF file and I will go to Cancer Genome Interpreter. So now you have two ways to actually annotate your file. So uh, the first one is if you will go to add file, and then you will select the VCF file you want to annotate, and you will open the file. So it will look like this. So you don't need this window at all because you already uploaded the file. And because in this window you can actually paste your mutation. For example, if I go to this file, like I already duplicated my VCF file, but this time I generated a Excel sheets. Because if I will open this file and then I will copy paste the chromosome position on the chromosome, and I will ignore the ID. I don't need this ID. I will just copy paste chromosome position, reference base and altered base, and then I will copy paste these details into that window. like this, copy, then you will go back to Cancer Genome Interpreter and then you will paste it like this. But I already have my VCF file, so I don't need to copy paste anything here. Another interesting thing is if you are confused how to paste manually your mutations, so they have actually given you all the formats that how you're gonna copy paste your variants. So I was basically copy pasting in this format, which is this one: chromosome number, position on the chromosome, reference and altered base. So I was pasting in this format, but you can paste in other formats as well. And now I will go down and I will select the cancer or the tumor of your sample. So basically, for example, if I have worked, for example, my VCF file is from breast cancer, so I will select breast. So we have different type of breast cancer, so you will do like this, but in my case I have worked with, for example, if my VCF file is from glioma, we will go down there what type of glioma it is, so you will select that one. So I will go for glioblastoma. So you have already selected glioblastoma, and then I will select the reference genome. So in my case my reference genome was HG19, and I have selected everything, and then I will press run. It will take time, like 1 or 2 or 3 minutes, and sometime more than that. But yeah. Okay, so I got my results, and here if you see, you can change the name of your sample. For example, in my case my sample was 0065. Okay, and then I have changed the name of my sample, and the cancer type I selected was glioblastoma, and the reference genome was HG19, which is correct, and total mutations was 157, and the driver mutations identified in this tumor sample is 19. So if I go here, they have nicely annotated driver mutations and passenger mutations. So I will, so input is basically a sample ID. So I haven't mentioned any sample ID, so that's why they have put input 01, but I haven't put any sample ID here. So that's fine. So I will go there, everything is okay, and then I will download the results. Okay, so I have downloaded these results, so I will right click and I will extract these files. Okay, so if I open it, so now you have 3 files. So one of your files is alterations, means your mutations, so you can't see it properly here. So if I select all the information and paste these information in my Excel sheet, I think it is easy to see everything here. So they have nicely telling me this is what I have given to CGI, and again, so these are all my mutations, and this is the gene of mutation, and this is the protein change, and also CGI is telling me what type of mutation it is. For example, this is non-protein affecting, and again, CGI oncogenic prediction is non-protein affecting, and this is the database ID for this mutation, and they have nicely given you extra annotations as well, and whether your mutation is on plus strand of DNA or minus strand of DNA. So CGI has given you a lot of information. So this is how you can annotate your variant file, but you have other files as well. For example, biomarker file. So if you open this and I select everything and paste in Excel sheet. So now basically this file is telling you the information about your mutations that you got in your sample, and biomarkers which is present in your sample as well. So this file is useful file as well for biomarker study. And input file is basically telling you what you have given to CGI for the analysis. So yes, this is what VCF file, this is the VCF file I added in CGI. And then the summary file is just the summary of analysis. So for example, the analysis number was this, and it how much time it took, which CGI version we have used so far, how much driver mutation we got. So Cancer Genome Interpreter is basically a useful tool, and especially for cancer studies. So I hope you enjoyed the video, and I hope I explain everything well. In case of any question, please put your question in comment section, and I hope I will help you with that. Thank you.